3B. For today's video, our topic was all about the microbial metabolism. As we all know that microbial metabolism is the process of catabolism and anabolism. The ATP productions are the adenosine triphosphate, a preferred source of useful energy for all cells. Breaking bond of third phosphate releases ideal amount of energy. Bond is easily broken. This is why organisms convert food energy to ATP energy. How ATP is produced? In most organisms, energy from a food source is converted to energy in ATP by glycolysis, followed by one of two processes, which is the fermentation or the low ATP yield, or the respiration, the high ATP yield. The electron transport and chemiosmosis occurs in the mitochondria of eukaryotes and at the plasma membrane of prokaryotes. The oxygen is usually the final electron acceptor, but other molecules can play this role in anaerobic respiration. The lipid and protein catabolism. The lipid and protein can also be used as sources of energy to produce ATP. The different amino acids enter glycolysis or the Krebs cycle at various stages. The fatty acids are broken down to acetyl groups and feed into the Krebs cycle. To sum up the overview of aerobic respiration, first, the glycolysis, which is a glucose or the 6-carbon, is oxidized and split into two molecules of pyruvic acid into 3-carbon. The Krebs cycle is the process of pyruvic acid and generates 3 carbon dioxide molecules. The third is an electron transport chain that accepts electron, NADH, and FODH that generates energy through sequential redox reaction called oxidative phosphorylation. Factors affecting enzyme activity. Temperature. Reactions occur more rapidly as temperature rises. As long as enzymes is active, the heat can denature enzymes. pH. The enzyme structure depends on pH. pH affects charge of R group's protein structure. In substrate, the reactions occur more rapidly as substrate rises. The saturation occurs when substrate is high enough. In enzymes denaturation, enzymes are polypeptides that retain their ability to function only when folded properly. Changes in temperature, pH, or salt can disrupt amino acid R group interactions, causing the protein to unfold and become denatured. Some enzymes require cofactors. Can be a metal ion, vitamin, and other non-protein. If the cofactor is organic, it is called a coenzyme. The enzyme is inactive without cofactor. Enzyme inhibition. Inhibitors bind enzymes in one of two ways. The competitive inhibition, which is the binding to active site. The allosteric inhibition, binding elsewhere, changing its shape. The inhibitors can bind reversibly, can come off, or irreversibly don't come off. Example, the poisons. Feedback inhibition. The end products of metabolism pathways are important reversible enzyme inhibitors. Inhibit first enzyme in pathway turning the pathway off. Can be competitive or allosteric inhibition, provide an important way of regulating end product levels. Respiration. After glycolysis, energy and pyruvate and NADH is used to produce much more ATP by respiration. In the Krebs cycle, the breakdown pyruvate to 3-carbon dioxide, energy captured as electronegative by NADH and FADH2. In electron transport, the electronegative from NADH1, FADH2 are used to produce hydrogen positive gradient. In chemiosmosis, the hydrogen positive gradient used to make ATP. In earliest prokaryotes, most numerous organisms on Earth, it includes all bacteria. The earliest fossils dated over 2.5 billion years old. Autotrophic processes. All organisms depend on autotrophs. Autotrophs can produce organic molecules from carbon dioxide, an inorganic carbon source. All heterotrophs requires an organic source of carbon. The organic molecules directly or indirectly came from autotrophs. The source of energy for autotrophic processes can be light, which is the photoautotrophs that can carry out photosynthesis. The chemic autotrophs that use various molecules as a source of high energy. The light reactions of photosynthesis. Electrons from water energized by sunlight. It fuels the synthesis of ATP through electron transport and chemiosmosis, much like respirations. The ultimate reduce of NADP positive to NADPH, the ATP and NADPH provide energy to foil productions of sugars in the dark reactions. The dark reactions involves an anabolic pathway known as a Calvin-Benson cycle. Endergonic reactions of this pathway are foiled by ATP and NADPH from the light reactions. Resulting sugars can be used as a source of energy to build other organic molecules. Glycolysis. The glycolysis is a catabolic pathway by which sugars as glucose and several other food sources are broken down to 2,3 carbon molecules of pyruvic acid or pyruvate. It releases energy to yield 2 ATP per glucose. Also transfers high energy electrons, which is the H plus, to NAD plus to yield 2 NADH. All living cells depend on biochemical reactions to maintain its homeostasis. All the biochemical reactions in an organism are collectively referred to as metabolism, which has 2 basic types. These are the catabolism, the process by which a living organism obtains its energy and raw materials from nutrients. The anabolism is the process by which energy and raw materials are used to build macromolecules and cellular structures. Most biochemical reactions are part of a series of reactions referred to as a metabolic pathway. It usually takes multiple reactions to make end product. The pathway can be catabolic or anabolic. Each reaction is catalyzed by its own enzyme. Almost all biochemical reactions are catalyzed by a specific enzyme. The proteins that accelerate the rate of reactions without being changed themselves, to lower the activation energy. The needs of enzymes provides a way to control or regulate biochemical reactions. Reactions won't occur unless the enzyme that catalyzes the reactions is present and active. The oxidation and reduction. Much of the energy in food molecules such as glucose is captured as high-energy electrons by electron carrier such as NADH and FADH2. When a molecule receives or gains electron, it is said to be reduced. Electrons are typically transferred as part of the hydrogen atom. A molecule that gives up electrons, loses hydrogen, is said to be oxidized. Fermentation. The ATP production begins and ends with glycolysis in organisms that ferment. Fermentation is all about recycling NAD positive so that glycolysis can continue. The NADH is oxidized to NAD positive by reducing pyruvate to lactic acid, for example. The Krebs cycle, a cyclical metabolic pathway catalyzed by enzymes in the matrix of mitochondria. It requires two carbon acetyl groups connected to coenzyme A, which is the acetyl-CoA. As we all know, when we see prokaryotic cells, we are referring to cells that do not have a nuclear membrane. That is the reason why, if you will be looking at the structures of the nucleus, they do not have nucleus. Instead, they have a nucleoid, where in the genetic materials are nearly suspended to the cytoplasm of the cell.